All right, a couple of quick announcements really fast for today. One, we have a student who is trying to form a study group. This student, by the way, just not to toot the person's own horn, has done very well on the exams. You would do well to meet up with this person and want to study with them. I'm just saying. And don't be shy and think, well, no, I don't want to study with somebody that's doing awesome on exams, okay? Everybody benefits when they do a study group and stuff. So that person's going to wave their hand right now. They have also posted online on the 227 blog about wanting to form a study group. So, because I don't say names because we're recording and stuff. So go on the website, go in there, see the post about wanting to do a study group and meet up with that person. I'm telling you guys, it'll be well worth your time for everybody. This person wants to do it. You guys should do it. Find a time that works. It's awesome. All right. Second thing I want to mention, and if somebody will email me later so I don't forget in case I do forget, today only, there is an app called Visible Body. So if you type in Visible Body, and then it's the Human Anatomy Atlas 2018, complete 3D human body, blah, blah, blah. It's only 99 cents today. You could use it now. You could use it for 228. So Sherry is letting all the 228 students know. We'll put I think she'll put a post on the AMP website, and then I will try to remember, and if I have forgotten, somebody email me after class. Remind me to send you guys an email. I'll just shoot a general email to all the 227 students saying, hey, it's $1 today. It's today only. So if you guys get a chance, do that. It's Visible Body Human Anatomy Atlas 2018 for $1. So you can pull up stuff for 227 for muscles this time around. You can do stuff for 228 for next semester. And $1 is really good because if you look at a lot of the other apps, they're $20-something, $30-something, that kind of thing. So $1, pretty good. All right, the other thing, let's see, we are working on nervous physiology. We had just gotten done talking a little bit about EPSPs, or excitatory, presynaptic, postsynaptic potentials, IPSPs, inhibitory, presynaptic potentials, postsynaptic potentials, right? Which one is it, pre or post? I don't want to say it wrong. Postsynaptic potentials, that's right. Okay, so I told you guys that I would draw a picture in a second. And remember, it's a summation by the postsynaptic neurons. So just really quick review of the whole nervous physiology. You should make your own little notes. Draw like a little quick bullet points. They don't even have to be notes like this, but bullet points. So generation of an action potential. This is a review of the last 2 lectures, essentially. When a neuron is in a resting state, all the gated channels are closed, correct? All of them are closed. Gated channels, that's both chemical as well as, what's the other word? Chemical as well as voltage, right? Everything is closed when it's resting, okay? And those are the gated channels. Leak channels are still leaking stuff, right? Remember, we are leaking. But all gated channels are closed resting. So that means sodium gates are closed, potassium gates are closed. When a neuron depolarizes, it means something happened at that dendritic or receiving end. The neuron experiences something, and that's called a graded potential. So for example, when we were talking about our muscle physiology, what was the neurotransmitter that was dripping on that neuromuscular junction? ACH. So acetylcholine was dripping at the dendritic end. So if acetylcholine comes into contact at the dendritic end of a neuron, okay, what gates would open? No. So this is a chemical gate, right? Acetylcholine for a neuron is dripping on there. What gate opens on the dendritic end of a neuron? The sodium gates. So this causes the graded potential, which are small depolarizations, right? And so for the examples we started talking about here with our synaptic integration, summation of the postsynaptic, oh, I didn't put the thingy back on for you guys, sorry. So with the postsynaptic, thank you. For the postsynaptic neuron, we are dripping small amounts. This might be 2 millivolts or 5 millivolts. So for the examples I'm going to draw in a second, we'll keep it simple and easy. We'll do 5 or 10 millivolts of drippage amounts, okay? So which neuron, which means if sodium is our positive ion that's going in, then if we are going from -70 and you're dripping a positive 5 millivolts, say, at a time, where are we going from -70 if we get one amount, let's say, one bolus of an amount? One drippage let's say if it equals 5, what is now our negative 65? Okay, so you were negative 70 for a resting membrane potential. If we drip a 5 millivolt amount and sodium is a positive ion, that means we're dripping a positive 5, which brings you to negative 65. It's bringing you closer to threshold. But remember, graded potentials are not enough. One graded potential is not going to be enough to get you to that negative 55, correct? So remember, old exams might say 50, so just keep that in mind. And so the numbers might be off a little bit. So before it would take 20 millivolts, but it really now only takes 15 millivolts to make that change. So newer research, newer information, right? And then if we dripped another 5, so we went from negative 70 to negative 65, we drip another 5 acetylcholine's worth in there, so it opens up the sodium-gated channel. It allows sodium to come in for another 5 millivolts worth. Now where are we at? Negative 60 millivolts. All right. So we have graded potentials of, say, 5 or so. Is that going to be enough to cause the axon to fire? No. Okay. So what if we add some more neurotransmitter, like 10? So if we went negative 70 to negative 65, now we add another 10. Now where are we at? Negative 55. So then, yes, now we can hit threshold. Now we will get an action potential. That'll start the rest of our domino effect, okay, that we had talked about. So if we get to negative 55, a 15 millivolt change, then that will travel to the axon hillock, right? That's our important area of the axon. So the axon hillock feels the graded potential, okay? And that now does what? It opens up all of those sodium gates, okay? Let me make sure my doodad works. So this area is the axon hillock, right? We had our graded potentials are occurring over here. It then is enough from negative 70, we hit negative 55 at the axon hillock. It will then open the sodium gates. This is your domino effect. Ding, 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 ding. Open, 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 open. All the way down. That's why it does not matter how long this axon is. It doesn't matter if it was a meter. It doesn't matter if it's infinity long. It's going to zip all the way down that axon. Okay? And then, if sodium is now rushing into your cell, now what do you think the voltage inside of the cell now will be? It's becoming more and more positive. So it's going from negative 55, and in this case, when our voltage inside the cell hits a positive 30 millivolts, so that's from negative 70 to positive 30 is a 100 millivolt change. So when we hit positive 30, what happens then? This is a voltage sensitive gate, for 2 gates actually. Positive 30 means that sodium gate that's just after the axon hillock slams shut because it's voltage sensitive. If you hit positive 30, it closes. Okay? Now if you hit positive 30, sodium gates close, but what other voltage sensitive gate opens? Potassium gate opens. So here you have, if you want to think about it, negative 55 sodium opens all the way until it becomes positive 30. Sodium slams shut. Thus, potassium opens. Okay? So it's kind of like a multi-domino effect occurring. Dominoes are going in the sodium direction, ting, 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 ting. But then they also have like a second layer of dominoes on the potassium section going open, 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 open. So open, close, open, close, open, close, open, close, open, close, open, close. Because you have negative 55 all of a sudden shoots up to positive 30. Positive 30 means that gate now shuts closed. But the potassium gate that's also on that membrane opens up. And then you have the other sodium that then slams open because he now feels that action potential of the from hitting threshold. So he opens. So that's why it's open sodium, open sodium, open sodium. But as soon as you hit 30, that same sodium now closes, 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 closes. And as you hit positive 30, then all the potassium ones start open, 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 open. Okay? So it happens like little tiny areas all along this axon hillock. Okay? Remember, if it's a myelinated neuron, okay, these sodium gates and those potassium gates that are voltage sensitive are in between. They're at the nodes of Ranvier. Because where you have all that myelin wrapping around it, it's insulating it. You don't have anything, nothing can get through in that area really. Because you have like a cellophane wrapping all around it that's really a fatty layer, right? So all those sodium sensitive gates are in the in-betweens, in those nodes of Ranvier. So the myelin sheath area keeps everything protected and doesn't allow things to attenuate or decrease, so you don't lose that charge amount. And then you get to that non-myelinated area where you have all these sodium and potassium gates. So they get the signal, so all of a sudden they start open, 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 and at the same time close, 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 and the potassium's open, 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 open. Does that make sense? And then you hit another myelinated area, so then it leaps across again. This is that saltatory ching, ching, ching. So we've been learning about all these pieces separately, but when you guys go to study, put all the information together and think about it. Is this a myelinated neuron? What would it look like? If this is a non-myelinated neuron, it's going to look like this example here that we drew before. But if it's myelinated, it goes back to these pictures here where it's kind of showing you these are the non-myelinated ones have a continuous one where it's that domino effect, ching, 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 ching. And you still have a domino effect on the saltatory ones that have myelin. It's just you also get to skip parts, and this is what allows things to travel faster. Okay, so I just wanted to kind of review all of that. So asking just a couple of last questions before I draw those pictures real quick when we're talking about this. So when I said, okay, so all the sodium gates open, that caused the action potential that goes all the way down the whole length of the axon. So what caused all the sodium gates to open on the axon? What was the trigger? This is on the axon. No, that's on the dendritic end. That's on the body. No, that's the same thing the other person said. Voltage change. Voltage change, because axon is voltage, okay? And the chemical gated is on the dendritic end, on the soma, on the body of the cell body for the neuron. Axon is always voltage. So you have two different gates working here, okay? So let's see what else do I want to tell you. I think I told you everything. Yeah. Oh, wait, I have another picture here. What do I want to say there? And so what opened the sodium gates on the dendritic end? You guys said the neurotransmitter, or ACH in this case. And we're going to learn there's other neurotransmitters that we can use as well. But so we have two different types of sodium gates. We have a sodium gate that on the dendritic end that responds to neurotransmitter. So that's a chemical gate. And we have sodium gates that are axonal and respond to voltage change, okay? So that's a voltage gate. So we have graded potentials that travel to the axon hillock, and when we have enough depolarizations occur, threshold is reached. Threshold is what? Negative 55 millivolts, all right? And it's positive feedback, which is self-propagating. It's that whole domino effect. Once it starts, it's all or nothing, correct? All right. And what else do we want to say? Once we had, what were the potassium gates on the axon sensitive to? What allows them to open? Positive 30 millivolts, which means it's a voltage sensitive gate. Okay, good. And when we get that hyperpolarization part, that little dip, right? What does that do to? Do you guys remember? This is all review. Say it a little louder, I can't hear.
Potassium gates close really slowly.
Yeah, so the potassium gates close, but they close more slowly. So they hit that negative 70 and they start to close because that's its voltage sensitivity to close the gate. But it doesn't close like it doesn't snap shut super fast. So it slowly closes. So more potassium is leaving, which means you become even more slightly negative, thus hyperpolarized. So you're even a little bit more further away from threshold. So you went from negative 70, which is your resting membrane potential, to maybe negative 72, or maybe you're negative 75, whatever that might be. So you would need to get to 55, the negative 55 for the next one. If you wanted to re-stimulate that nerve during that time period, you would need more than that 15 millivolts, correct? Okay, so if it was negative 72, you'd need 17 millivolts to get you to negative 55, okay? So I'm playing with the numbers here because, one, I want you guys to understand it, but two, if you're looking at old exams, I don't want you getting confused by the correct answer back then if they thought it was negative 50 versus now it's negative 55. So just keep that in mind when you're looking at that. And what do we do to get it from that a little hyperpolarized area back to the negative 70? What's going on in that part? Sodium potassium pumps cost us an ATP. Okay, so what are we doing? We're pumping 3 sodium out with 2 potassium in. Costs us 1 ATP to do it. That gets you back, because of those leak channels and everything we just did with depolarization, will get you back to the negative 70 millivolt resting membrane potential. Okay, so remember that's also talks about, you know, the absolute refractory period. Absolutely, you cannot what? You can't trigger another action potential because it's already in the middle of its action potential. You can't trigger it. Absolutely is not going to happen unless it's an absolute refractory period. But then we had a relative refractory period, which is when sodium is now coming back into the cell, or you're hyperpolarized, and you could potentially trigger another one if the stimulus is strong enough. You could do that. Okay. All right, you guys are now experts on that. Yes? Yeah. Yay! All right, so let's draw a diagram of some of what we had been talking about there. So I'm going to roll it up just a little bit. So if you have your, we're going to do a very simplified drawing. We're not going to stick all the dendrites, the crazy hair sticking off of it, but the body of it is our dendritic end, okay? And I'm not drawing the axon hillock, but you know the axon hillock is right off the body. Here's our axon, and here's an axon terminal. And so that would be, and then if we draw another one, okay, then this cell number 1, let's say, is our pre or postsynaptic cell. Pre because it's before, pre. So the number 2 cell is our postsynaptic cell, okay? So what we're saying here is, if, let's say, we have gotten some sort of stimulus, the brain is saying, I want to do something, or you have sensed something, and this isn't really talking about whether we're going to the CNS or from the CNS, but this could work in either direction. And I want you guys to realize that. That's why we're not saying specifically where we're going with this. But some stimulus occurs. And let's say it's worth about 5 millivolts, right? So if this is temporal summation, this neuron number 1, the pre-neuron, could hit rapid fire 5 times, 5 millivolts at a time, to make number 2, the postsynaptic cell, fire. So what that means is if I only get 5 millivolts worth, what's going to happen to number 2? Nothing, because that's not enough for number 2 to hit his threshold. If I say I'm going to rapid fire temporal summation and I hit it 1, 2, 3 times, now will I get anything? Yes, because 5 and 5 and 5 will give me 15. Negative 70 to plus 15 now puts me at negative 55. This cell can now fire and send his signal. And remember, this is a one-directional thing only, okay? This guy sends the signal that way. Number 2 senses the signal. If it's enough, he'll get his action potential and he will send it down along and he fires, okay? So I could have temporal summation do that. I could do something called spatial summation, which is many neurons firing at the same time. So let's say I have neuron number 3, who also happens to be a presynaptic neuron, okay? And number 3, if, let's say, so I'm going to take off my check mark here for a second and my arrows. So if number 1 sends 5 millivolts and number 3 sends 5 millivolts, okay, they each just fire once, is number 2 going to fire? No. Let's say, though, that, come on, let's say, though, that number 3 is worth 10 millivolts. So 1 fires and gives you 5 millivolts worth. 3 is firing, gives you 10 millivolts worth. Now will number 2 fire? Yes, because it's an additive effect. So these are a spatial summation example because it's different neurons firing at the same time and stimulating number 2. As long as number 2 gets to negative 55, you will get an action potential and send the signal down. Okay? So let's say now, what if I have another neuron here, we'll call number 4, okay, and let's say that this one is delivering negative 5 millivolts worth, okay? Looking at this information, what is the difference between 1, 3, and 4? What would you call number 1 and 3? An EPSP or an IPSP? So EPSP says excitatory neurotransmitter binding causes depolarization. An IPSP is a neurotransmitter binding causes hyperpolarization, meaning further away from that threshold, from negative 55. So if I wanted to give a name to what number 1 and number 3 are doing, they would be what? Say it again. EPSPs. Okay, because they are bringing us closer to threshold. They have positive, where our resting membrane potential is negative 70 millivolts. And if we're adding a positive 5, that means that puts you at a negative 65. And if number 10 is a positive 10, that put us now at negative 55, because we add them all together. And so what would number 4 be in comparison to 1 and 3? Right, an IPSP. So it shouldn't matter on an exam whether I give you a negative number or if I use the term IPSP or EPSP. If you're seeing positive numbers, you should automatically think that should be bringing you closer to threshold. If you see a negative number, it should make you think, okay, I'm getting further away, I'm hyperpolarizing, okay? And I said that it is a summation by postsynaptic neurons. So it means EPSPs plus IPSPs added at the axon hillock of number 2 hope to reach threshold. Thus we'll get a firing. So if number 1 fires once with a 5 and number 3 fires once at a 10, but number 4 is also firing at a negative 5, do we get an action potential? Number 2 will not fire, because what we did there was we did 5 plus 10 minus a 5 gave you 10, and 10 millivolts was not enough to reach negative 55. So what do we have to do here on this picture to make it happen? How do we make number 2 fire? Let's say number 4 stays the same. We could fire number 1 twice, right? And that would be a 5 plus 5 plus 10 minus a 5. That'll bring us to 15 millivolts. And then we would get our action potential, and number 2 would fire. So you guys can play with those numbers a little bit and see how many times would this guy have to fire. And think of it like little drips, right? Okay. Any questions on our pictures there? Okay, hopefully that makes sense and is easier to play with the terms. Play with the terms EPSP, IPSP.
What is number 2 usually?
Number 2 is the postsynaptic neuron. So 1, 3, and 4 were presynaptic neurons that are stimulating or hyperpolarizing, if you will, number 2. Sorry, I didn't hear that part.
So negative 70.
Yeah, negative 70 is the resting membrane potential. And so everything that 1, 3, and 4 are doing will affect that negative 70. And that's why we say it's a summation. You have to add all of those presynaptic neurons and what they're doing up together to see how it will stimulate number 2, which was the postsynaptic neuron. Okay? All right. Now, let's see, this picture down here, what are we saying? Okay, so this is just showing you the term spatial summation. This one is the spatial. This one is temporal. Remember, temporal is one neuron that is firing many times, which is all of the stair steps here. It's one going tink, tink, tink. It's dripping its neurotransmitter on cell number 2 here. And so if it's a single neuron doing it, that's temporal summation. And then it's showing you once you get enough to hit negative 55, you'll get that action potential, which is what the big blip is. On spatial summation, that was where we were saying that this is multiple neurons, could be 2, could be 3, could be however many it needs to be, but it's more than one firing at the same time. So it's hard to see on the picture, but they're saying this is neuron 1, neuron 2, neuron 3, neuron 4, neuron 5, or maybe neuron 1 hits you twice, but there's still another neuron after that. And again, once you hit negative 55, then you will get your action potential, the big blip. Okay, so the picture down here is showing, here's a neuron coming in, here's a neuron coming in, stimulating. This would be like our neuron number 2 was from up above. This picture is showing you only have one neuron coming in, the presynaptic, talking to neuron number 2, the postsynaptic. So this one has 1, 2, 3, 4, 5 neurons not touching, but having a synapse with number 2, the postsynaptic. Does that make more sense seeing the picture now? Maybe?
Are the negative inclines on the graph or the negative impulse?
It could be, or it could be that it you hit, remember, graded potentials are very short-lived. So if, let's say, you have one of those neurons, let's say number 1 fires, and number 3 maybe doesn't fire at the same time. Maybe number 3 fires a second later. And so number 1's effect might have started to degrade already. And then, so that might be your little drop off, and then you have another stimulation. And again, you could have many graded potentials, but until you hit 55, nothing will happen. So that could be part of the drop off there, yes.
Looking for like leak channels or?
Could be leak channels, could be that you just didn't have another one hit close enough to be additive enough to get you to the negative 55. So it could be a couple different reasons. Yeah. Cool? All right. Now we can talk about some of these neurotransmitters. So there's approximately 100 known neurotransmitters, and more get discovered every day. So we've only talked about acetylcholine, but there are many others. We're going to talk about just a few of them. And it says 100 known neurotransmitters, many neurons make 2 or more. So you could have one neuron could make 2 or more different neurotransmitters that'll do different jobs, just depending. And when we get into 228, you guys will see that more on how we stimulate other things on the systems side of the body, if you will. And it says the stimulus frequency dictates which one is released. So stimulus frequency is like how many times are you firing or activating it to fire like number 2? How many times are you stimulating that number 2 might depend on whether 2 releases one neurotransmitter versus a different one. So low frequency might be one type is released, and maybe if you're really stimulated and you're like, hey, pay attention here, ding, 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 ding. I'm sending lots of neurotransmitter on this one or gated channel, and then this guy over here says, ooh, we really need to do something. I'm going to send out 2 different neurotransmitters. That's pretty much what it's saying. We don't really go into a lot of detail with that, but so it's self-modulating. So not everything has to communicate to your brain for your body to do some of the things that it does. It does it on a local level sometimes, okay? So let's look at, we can classify our neurotransmitters by a chemical structure or by its function. So we'll start with chemical structure. So acetylcholine, ACH, you guys already know. You do need to memorize these neurotransmitters and how they're classified and be able to play with how they work. So know them by their structure or know how something would function as excitatory or inhibitory, and we'll talk about that in a second. So acetylcholine, we know, was delivered to all skeletal muscles, right? We're stimulated by this. And little m is my abbreviation for muscle, and that was at the neuromuscular junction. Okay, and some autonomic systems, so some of our parasympathetic and sympathetic will be stimulated by acetylcholine. So some autonomic, okay, and also some parts for the brain will be stimulated by acetylcholine, okay. The next category or chemical structure are what are called biogenic amines. So this means that you have one particular, in this case, a monoamine, so a single amine or amino acid. We talked about amino acids before when we were looking at early structures, right? So that it has what's called a parent compound, which in this case, it's saying like the original compound that we're talking about is a monoamine, meaning mono is one, so one amino acid, which ends up getting modified into the actual neurotransmitters that we're interested in. So NT for neurotransmitters. And so the main one that we'll talk about is tyrosine, is an amino acid. And it's a biogenic amine because it gets then converted into, it's a precursor to the neurotransmitter. So it gets changed into, we can have epinephrine. It can make norepinephrine. And it can also make D for dopamine. Okay. Another biogenic amine that we have that can be converted into a neurotransmitter that we use is tryptophan. Okay. You guys are somewhat probably familiar with tryptophan. People like to make jokes about it around Thanksgiving time, right? Your sleepy thing. So when you eat tons of turkey, which has tryptophan in it, milk, when you're having a hard night getting to sleep, you might warm some milk up and drink some of that. Also has tryptophan in it. So this is an amino acid that can then get converted into dun, 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 serotonin. I'm going to put my arrow over here because I ran out of room. Serotonin. Okay, so serotonin is the neurotransmitter. Tryptophan was the amino acid, the precursor. So the other way that you might see serotonin written is also HT. Okay, so 5-hydroxytryptamine. You might also see it as 5-HTP for 5-hydroxytryptophan that then gets converted into serotonin. So there might be many steps sometimes for conversion, but the idea of biogenic amine, you have an amino acid like tyrosine or tryptophan that then gets converted into the neurotransmitter that will then stimulate the next neuron, if you will, right? So we have tyrosine can become epinephrine, norepinephrine, or dopamine. Tryptophan can become serotonin. You can also have amino acids that will act as neurotransmitters in and of themselves. They don't have to be converted into something else. So for example, there are a few amino acids, such as the two I want you guys to know, is glycine and glutamate. Okay, so let's see. So the difference is a biogenic amine is an amino acid precursor that has to become the neurotransmitter. In this case, if it's just the amino acid itself is the neurotransmitter. So we have glycine is an amino acid that is also a neurotransmitter, and glutamate. Okay, and those are only stimulating or in the CNS. So some people, for example, anybody sensitive to MSG, monosodium glutamate. So some people can have MSG in their food or drinks or sauces or whatever, and it will not bother them. Some people might be sensitive to it, and it is a neurotransmitter. And so some people might get headaches or migraines from food that has glutamate in it, such as MSG. So I'm just giving you guys info, things that you can help relate to things that you might have heard or understand for yourself. We can also have peptides, okay, or you might also hear it called neuropeptide. What's a peptide? It's a protein. And if we say neuropeptide, we're saying a specific brain protein, a neuron type protein, right? So these are chains of amino acids, because that's what a protein is, right? Amino acids linked together. So chains of, and I abbreviate amino acids, little a, that act as a neurotransmitter. So in the CNS and PNS, okay, we have something that we call substance P. I know this sounds like a funny, like sci-fi kind of thing. Substance P. Anybody ever watch what was it?
Sci-fi named Substance P.
See, right? I always think of that movie. Oh shoot, what was it called? Oh, it was super cool with the bugs and darn it,
Big bugs?
had big bugs in it. Other planets. Yes, okay, well you guys know what I'm talking about, I think. Anyway, so they had like substance blah, 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 you know, from the bugs that, you know, anyway. So substance P, it is actually a real thing. Guess what P stands for? Not urine. It stands for pain. So substance P is for pain. So for example, they think that people that have fibromyalgia are more sensitive or have more receptors for substance P. Thus, they might notice pain where you could touch somebody that has no fibromyalgia and you could grab their arm, not like, I'm not saying like brutally grabbing somebody, but you grab their arm and like, hey, come with me over here, and it wouldn't bother them. But somebody with fibromyalgia, you grab their arm and just the physical, remember deformation on pressure could stimulate nerves. So that could be enough to stimulate their sensory neurons that then recognize substance P, and something that shouldn't have hurt is very painful to them.
They scream at you.
And they scream at you, and it takes them sometimes days or longer to recover from something that normally would not have affected that person. So you had people that are saying, I hurt, and nobody's believing them. Now we have a name for it, substance P, very scientific, right? But it actually is. So we have substance P, which is a pain signal mediator. So I abbreviate pain as PN. So pain substance mediator. Okay, so the neurons involved in pain pathways, if substance P is released, these people are going to definitely notice or be in more pain. We have natural opiates in our body also. Opiates as in painkillers, right? Natural opiates. So for example, you have people that we can all release enkephalons, okay? And we can also release beta endorphins. So enkephalons, and also this is the Greek letter beta endorphins. So for example, people that love to exercise, love to run, exercise 3 times a day because they are getting a high from it. Yes, they are getting a high from it, a natural high from it. Their body, it's kind of like when you're pushing your body to the point where you think you can't do it anymore, and then you get that second wind, and you're like, I'm ready to go again. Let's do this thing. And you're like, are you crazy? What is wrong with you? And you're like, I feel good. Let's do this, right? That's their beta endorphins being released naturally in their own body. They feel good. They are getting a natural high. And then some people supplement those natural highs by buying things, street drugs or through their local pharmacy, and can also give themselves a high that are not natural opiates, but they're based on opiates, right? Another natural opiate we said is enkephalon. So for example, how about women giving birth? Did you ever notice after, if any of you have had children, that after you've delivered, you're like, I could do this again, right? Yes, and you can. Okay. So that would be your body's natural way of helping you forget that was really painful a second ago. But your body will release it because, yes, we obviously can survive childbirth. There are obvious complications and things that make that not so survivable at times, but otherwise we wouldn't all be here, right? Yes, okay. So natural opiates, enkephalons are like with labor. Beta endorphins are like your runners high, your exercisers, those second wind people. And these help get rid of pain, right? You don't feel pain when this stuff is released. You feel pretty good. So we can also, so those we're classifying them by their structures. What are they, right? Now let's classify them by their function, okay? How do they work? So we could have excitatory and inhibitory. So, or we should say excitatory or inhibitory is what we should say. So this is based upon what neurotransmitter is released. Will depend on whether it is excitatory like an EPSP or inhibitory like an IPSP. So this is based on neurotransmitter and location, right? So what might be an excitatory neurotransmitter for one area of the body or receptor could be inhibitory somewhere else in the body for a different receptor. So that's why it says whatever the neurotransmitter is could determine if it's a positive or negative effect or excitatory versus inhibitory, or where is it stimulating something, skeletal muscle versus somewhere in the brain or something like that. So for example, acetylcholine, oh look, there's that guy again. He is, what do you think he is to skeletal muscle? Excitatory or inhibitory? Excitatory, yeah. So we have excitatory for skeletal muscle, but it's inhibitory for cardiac muscle. Okay, so when acetylcholine gets released by the parasympathetic system, what does parasympathetic do for us? Rest, digest. Because sympathetic is fight or flight, right? The bear is chasing me. I just have to run faster than you, right? Or be able to do something to prevent the bear from eating me. So parasympathetic is rest, digest. So is your heart rate going to increase or decrease if it's parasympathetic and you should be resting, digesting? What should your heart be doing? Resting, relaxing. And it still has to pump at a normal rate, but it's not going to be, it's not pumping super fast because you got to get more blood to your legs and you got to get out of here, right? So parasympathetic means that it is inhibitory on cardiac muscle. Okay, so let's see. How about indirect acting? Or sorry, direct acting. I'm trying to skip for you guys. So direct acting means that the neurotransmitter binds to receptors, which will open ion gates. So we've already seen that once, right? Neurotransmitter, for example, acetylcholine, bound to the motor end plate of the skeletal muscle cell, which cause the sodium ion gates to open. That's exactly what that's talking about there, okay? So when you guys are studying this, go back and use the examples that you've already learned and apply them to the new terminology that you're learning and see how that plays out. So for example, that was acetylcholine opens the ion gates. This is a short effect, usually with direct acting. Short effect, that's a T, okay? So that just means it's not super long lasting, okay? And that's why we had to keep releasing acetylcholine if we wanted to keep that muscle contracting and holding its contraction, right? If we look at indirect acting, that means that the neurotransmitter binds, or when it binds, will start a cascade of effects. So neurotransmitter binds, but it causes a cascade of effects. Cascade literally means like, if you're thinking of a waterfall and the water is hitting all these rocks on the way down, so it's like tink, 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 tink. It's causing multiple other things to have to happen, a cascade of effects. One thing happens, then this thing happens, then the next thing can happen, then the final product happens. That's what we're talking about. So that cascade of effects inside of a cell and that will cause it to function longer. So this actually, a cascade of effects will have a longer lasting effect. So longer effect, which I know sounds funny, indirect, longer effect. So just think direct, like when somebody is being very direct with you, they're very short with their answer, right? Short and sweet and done. And think indirect, it's like, well, you know, sometimes. So if you talk to Sherry, you will get a very direct answer. If you talk to me, I tend to be indirect. And I'm like, well, that's okay, but you know, if you try this over here and then you do it, Sherry'd be like, did you study? Next, you know, so think of it that way. That'll help you guys remember. All right, very good. Direct is not bad, okay? I'm not saying that's a bad thing. That's a good thing. I like it when people are direct. They tell you what you need to do and get it done, right? Okay, so acetylcholine, you know, we keep using this guy as an example. So if I were studying for the exam, I would like figure out everything I could about acetylcholine and all the different ways that it might function, direct, indirect, excitatory, inhibitory, chemical versus function, classification, that kind of thing. So acetylcholine can be classified a few different ways, by its structure and by its function, okay? Like, you know, if it's direct acting, and is it excitatory, inhibitory, and the location that it goes to, you know, is it going to cardiac muscle, was it going to skeletal muscle, that kind of thing. So, dun, 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 neural integration makes possible complex behaviors. That sounds weird. Neural integration makes possible. Complex behaviors are possible because of neural integration. I like that better. Yes, let's do that. Neuronal pools. So, functional groups of neurons creating circuits or pathways. That's the key here, circuits and pathways, okay? So, integration, remember we said we had interneurons, like a neuron in between the other ones, right? So that's what you kind of want to think about here. And we have our 4 pictures and our 4 types of neuronal pools, so, or 4 types of circuits, okay? So the basic types, the 4 that you guys need to know are converging, diverging, reverberating circuits, or parallel after discharge. That sounds kind of strange, but anyway. So let's first talk about them, and then I think it'll make more sense when you're looking at the pictures, okay? So converging, another name for that would be concentrating circuit. So if you're concentrating or converging, what does that sound like? You're like wide going smaller. So this is many neurons to one neuron, okay? So it says many to one relationship, so it's converging. So in the picture here, that's this first guy right here. So you have neuron 1, neuron 2, neuron 3, number 4, all interacting with the dendritic end of, say, neuron number, we'll just call it 10, so it's a totally different number, right? So these other 4 neurons are converging to one neuron, okay? The idea behind this is these are seen with your common sensory circuits or pathways. So common with, oh, my tummy's growling, sensory circuits. So many senses are being triggered to cause a single effect. Many senses triggered. There's an E in there. Causing a single effect. Okay, so for example, a really good one is bacon, right? So when you say the word bacon, you're hearing it, right? I'm saying the word bacon. Maybe you walk into a room, maybe you're smelling the bacon. Maybe did your mouth already start to salivate if you like bacon? If you don't like bacon, you're not going to salivate. But so what I'm saying is just even the word bacon, the association of the word for many of you caused your mouth to already get more saliva in it. Okay, like the Pavlovian thing. If you walked into the room and you see the bacon, you have an idea of what bacon means to you. Delicious. I think my tummy is growling. I want to eat it. It might be that you smell the bacon, and that could also cause you to salivate and get ready to eat and digest it. It might be that you hear the bacon sizzling while it's cooking. So that's another hearing aspect of it. So many senses just got stimulated if you actually didn't just hear it and see it, like walk in the room with it, but if you had, but even just saying the word you've already associated with it, and I already have like, I have to swallow more right now just because of saying that. Because have you not walked into a restaurant and it says bacon, you're like, I want to order that. And it may not even be good, but it said bacon. Come on, right? So that is what we're talking about. Many senses got stimulated for one effect. You're creating more saliva because you're getting ready to digest. Just the word, or any favorite food of yours that had a certain smell or whatever to it, if you think about it, you will already start getting ready because you know that means, ooh, food, let's get ready to eat. You're going to start digesting it here and then get your tummy ready for it, and you're releasing certain chemicals already just because I said the word bacon. So that's the idea of converging. Converging, so sensory, many senses get triggered, causing a single effect. If you were to draw a simplistic picture of the same thing, that's kind of like our picture up above, where you have one neuron here, another neuron there, another guy here. Let's pretend that his dendritic end to this guy here, okay? So many of them converging onto one. Diverging is the mirror image of this. So just think, flip it around. So they call this amplifying a circuit, right? So this is a one-to-many. So if you look at the difference between converging versus diverging, instead of many to one, you're now going one to many, right? So this is common with sensory and motor systems. So common with sensory and motor circuits. Okay. And this would be many neurons having to turn on in order to coordinate maybe an event. So walking, for example. It's not simply, I mean, you guys have forgotten what you had to do to learn how to walk. But if you guys watch a toddler that's just starting to learn and how awkward it looks, right? And now look at an older child or an adult and how coordinated they look when they walk, they make it look easy, right? And the little toddler's like, I want to get from here to there. And this is really hard. But you had to coordinate different things. You had to coordinate muscles, different groups of muscles to relax and contract in order to hold you upright and not just tip over. You had to coordinate balance with your cerebellum. You had to coordinate a lot of different things. So that's where you want to do one thing. Your brain says, I want to walk. And then it's telling many other neurons what it needs to do in order to allow it to look coordinated and get you from point A to point B. So walking would be a good one to think about that. Another one would be if you had a hot pot on the stove. Maybe you didn't realize it was hot. You didn't realize that you had already turned the stove on or how long you had left it on at that point. And you go to grab the pot because you weren't thinking about it. And you have one action. You grab the pot. And all of a sudden, two different things happen very quickly if it's hot, right? What do? First, you're going to what? Let go of the hot pot. Have you ever seen a kid, though, that grabs onto something hot and they don't let go, like, ah! They haven't quite coordinated yet. But anyway, they will. So immediately you let go of the hot thing because you know you got a sensory sensation, hot, let go, right? Don't touch me anymore. That hurt, okay? But you also had a pathway that also occurred that didn't even talk to your brain. You realized afterwards that it was hot. You let go of it before you realize it, unless you're that little toddler kid that didn't realize it yet. You're still making association neurons at that point. So that's the hot pot. You touch a hot pot, you have a withdrawal reflex, which the reflex, by the way, does not talk to your brain. It just does it. That's the let go part right away. And then at the same time, you have another signal going to the brain saying, OMG, that was freaking hot. Okay? So now you have the idea of what it was, what just happened. You have the sensory of that is hot. So you had two separate things going on at once, and that's what's going on here. You had hot coming in, and then many different things happened at that time. So if we drew a diverging, you have one neuron that then will talk to this guy over here, who might talk to this guy over there. He might have another collateral who might talk to this guy over there, and those guys can talk to somebody else or do whatever else that they're talking to, right? Then we can also have a reverberating circuit. So these are called circular pathways. So reverberating, you can just think it's kind of going on and on. It's a loop, essentially. Okay, so that's a circular pathway. So this, you just need to start the process, essentially. Okay, so just have to start the process. It can last seconds. It could last hours. It could last days. It could last years. It could last your whole lifetime. Okay? These reverberating circuits a lot of times have to do with memory and rhythmic activities. So a rhythmic activity might be breathing, sleeping, walking. Your heart starts to beat, and then hopefully it never stops until the day it does stop, right? Okay, so those are rhythmic activities. The process just had to get started, and then it just keeps going. Okay? So I have, can last seconds, hours, days, years, has to do with memory and rhythmic activities. I know my handwriting was kind of bad there, sorry. So you can think of it like turning on and off the system. So when you go to sleep, you're not really processing and thinking about, I need to go to sleep, I'm going to do it now. Sometimes we think that way and we're like, I can't fall asleep. If I could just fall asleep right now. But once you're asleep, things are on autopilot on your body, essentially, right? Until you wake up. So the sleep cycle would be a reverberating circuit, okay? If I were to draw that, that might be this guy communicating with this guy, communicating with that guy, and it just keeps on going. Stimulate, stimulate, stimulate the next guy, right? Until you have to stop stimulating it. And then the last one was parallel after discharge. And this one is pretty cool. This is one to several to one. So if you look at that first picture, they're saying, you know, one guy here is going to several, right? And then eventually they come back just to one again. So this is a diverging circuit to converging circuit, right? Does that make sense? Can you guys see that? Maybe. All right. So this is, the first half would be one to many. So that's your diverging. So if I were to draw that, that would be like this guy here. Okay. And he might, I keep wanting to draw arrows instead of my dendritic end. Sorry. Boop, boop. There we go. So he might talk to this guy, and this guy. And then they converge, those guys converge to one guy. So one to many and then many to one. Okay? What's important about this is one, it's described as an echo event. So it's an echo event. This is important because this is what really has to do with complex reasoning, not wine, Riesling. Complex reasoning, intellect, and your personality. Okay, so this is involved with problem solving. So this is higher order thinking, is what I'm saying. Do you guys see that, and does that make sense? So problem solving, like when you guys are learning complicated math, right? When you guys are learning A&P and you're trying to make the connections of something that I've said or we've written down in the notes and applying how it actually functions. And then when you guys are on the exam and you're trying to recall these memories and this information, your brain is going through all these different pathways trying to access all the other neurons, saying, how did I learn and process this and remember this, and then spit it back out and I can answer it on the exam or something, okay? So again, this is reasoning, intellect, your personality. It's not really clear why we have these guys here, but a lot of times it's because of all these extra interconnections, these extra neurons talking to each other, that makes us the top dog essentially on the planet, okay? Being able to do higher level reasoning, okay? Versus like an iguana. It's not that they're not intelligent in their own right, okay? But they do say dolphins are smart too, so.
There's a reason we're on the top of it.
That's what I'm saying, it's because of this, but anyway, so.
Kinda nice and not walking outside and worried some dinosaurs.
Right. I mean, if you were out in the Serengeti, you might have to worry about the lion or tiger or something. I would be afraid of Australia and all the other poisonous animals down there too, and critters. So, right? So they don't completely know why we have them, but because we have them. And this is where I was talking about, I think 2 lectures back, I think it was, where I was saying, when the difference between like children and their thought process, they are still growing and learning stuff, which we're going to see in the next chapter. They're making still a lot of these interneuron connections. One neuron growing to talk to another neuron over there and making a connection. Okay, and then remember when we said with some of the mental degenerative diseases where people become more childlike in their thinking and processing because they lose these connections. Okay? And you can have somebody that's 102 and doesn't lose those connections. But when you have somebody that has lost the connections, you can see a difference in their thought process and how they talk and think and apply their knowledge. And what knowledge can they recall when you've lost that? So that's what that's talking about. That's why I say this stuff is pretty cool when you look at it that way. All right, moving on to chapter 13. Dun, dun, dun. How did I go way down there? Go back to the top. Ta-da! Chapter 13, the brain and the spinal cord, meaning the CNS. All right, so we have 4 regions of the brain. We have the cerebral hemispheres. You have 2 of those, right? A right and a left cerebral hemisphere. You have one diencephalon. You have a brainstem, and you have a cerebellum. So the cerebral hemispheres are the bulk of the brain. Okay, that's the big mushroomy part on top of the brain. Those are your hemispheres. 1 and 2. Okay, you have a diencephalon, and this is not easily seen with the hemispheres attached on top because it's kind of like the mushroom cap that's plugged into the diencephalon being more like the beginning of the stem of the mushroom, right? You have the brainstem, which is composed of 3 different areas, and then you have the cerebellum, and that's where you can remember cerebellum has 2 L's or 2 legs in it. So this has to do with your balance and some of the coordination in that sense. So this is important for motor activity, essentially. Most of this should be a review because you guys already had your brain evals. Was that just last week, I think? Yeah. Yay! All right. So the other areas that we want to talk about are ventricles of the brain, which are the fluid-filled chambers in the brain, and they're lined with ependymal cells. Okay. So fluid-filled. What's the fluid inside these ventricles? CSF, cerebral spinal fluid, right? All right. And then they're lined with ependymal cells. Ventricles, thus, are these spaces or chambers. CSF is produced 24/7, constantly. There's a problem if it's not, okay? So CSF is produced 24/7 in each of the ventricles. So all of the ventricles are producing it, okay? Do you guys know what our ventricles are? We have 2 lateral ventricles. Then we have a 3rd ventricle and a 4th ventricle. So we don't call the lateral ventricles 1 and 2 because they didn't want to say the right was better than the left or number 1 is over here versus there. So you just have 2 lateral ventricles, which is essentially your 1s and 2 ventricles. You have the 3rd ventricle and then you have the 4th ventricle. So let's see, lateral ventricles, 1 in each cerebral hemisphere. So here's a lateral ventricle and here's a lateral ventricle. Here is your 3rd ventricle and down here is your 4th ventricle. So I'll put L for lateral and lateral. They're trying to show them kind of sort of cockeyed a little bit so you know that one's superimposed over the other there. So lateral ventricles, 1 in each cerebral hemisphere. They are separated. Do you guys remember what they're separated by? What are they separated by? A thin membrane called the septum pellucidum. Isn't that a cool word? Separated by the septum pellucidum. So if you guys, when you sliced your sheep brain in half, depending on if you weren't directly in the center, you probably were likely just slightly off to one side. One of those sides, you should have been able to see the septum pellucidum. And a septum's a thin membrane, a little different than the septum in your nose that people will pierce sometimes, but the septum pellucidum, okay? We are connected to the 3rd ventricle by the interventricular foramen. That's right here. So this allows fluid flow to go there from laterals to the 3rd. Okay. So 3rd ventricle is on the midline of the brain in the diencephalon. So 3rd ventricle is in the diencephalon. It is very thin. It's not very big. And it's connected to the lateral ventricles by the like in between, interventricular foramen. Okay. Then the 4th ventricle is behind the 3rd ventricle and just anterior to the cerebellum. So that's why he's down here. Okay, this guy right there. And so that's why they're saying he is inferior to the 3rd and behind it a little bit, so posterior would probably be a slightly better word, and just anterior though to the cerebellum. Here's the cerebellum. These are your cerebral cortex or hemispheres, right? Cortex is up top, cerebellum is on the bottom. I'll put 2 L's there so you remember cerebellum with an O. Later you'll look, what's C11? I don't know. All right, so 4th ventricle connected to the 3rd by the cerebral aqueduct anteriorly. So that's this flow right here. Okay, so that's your cerebral aqueduct, or you could put CA to remind yourself that, and attaches to the central canal of the spinal cord posteriorly. So SC is spinal cord, posteriorly. So CSF flows from the lateral ventricles through the interventricular foramen to the 3rd ventricle, through the cerebral aqueduct to the 4th ventricle, through the central canal of the spinal cord, and it can leave by these 2 little things called the lateral and medial apertures, which means like these little holes. So here's the lateral aperture and the median aperture. So it's like a little leak area there and a little leak area there, if you will. Okay, like little holes. Okay. So sometimes the cerebral aqueduct, which we said was right there, okay, so I'll put CA to remind you, cerebral aqueduct is right there. Sometimes it can get clogged, okay? So if the cerebral aqueduct got clogged, do you still get cerebral spinal fluid in the 4th ventricle? It does still have cerebral spinal fluid because every ventricle produces cerebral spinal fluid. So it will still have cerebral spinal fluid there. But if it gets clogged, what do you think happens above the cerebral aqueduct? What happens to 3rd and lateral ventricles? It could swell because it's still producing it, but it's not flowing down into the 4th ventricle. It got stuck up there. So if you're a little kid, and remember when you guys looked at the fetal skull, it still has areas that can shift on the skull, right? So if you had your ventricles filling up with more fluid but nowhere for it to escape, then this is like what we're talking about, hydroencephalus, encephalopathy. So hydro, water, people will just say, oh, it means water on the brain. Well, kinda, yeah, because most of your cerebral spinal fluid is a lot of fluid water, right? So fluid on the brain. But if you're filling up on the inside, what's happening to the brain cortex? It's getting mushed, it's getting compressed, right? Now, if it's a kid, their skull can allow for some filling up and pressure outward. Thus, you'll see like a picture, I think they even did a meme of that poor kid, where he had a very alien-like head, where it was very bulbous looking and strange, did not look like a typical child's head, right? If an adult had it, well, you're kind of fused for the most part with the sutures, right? So what do you think is happening with an adult who gets hydroencephalopathy? You're getting the pressure. You might potentially have brain damage depending on how long it can potentially be there, okay? Because if you're compressing on other areas and things can't function the way that they should, that could be a big problem. So a lot of times they'll do a shunt where they basically have to poke a hole, put a tube in it, and bring that tube usually down to the abdominal cavity so it can just drip into there and your body can reabsorb the fluid and do whatever it wants with it. And hopefully the cord is long enough, things shouldn't be going back up the other way. But if that clogging is there and it can't be relieved, because do you guys realize how medial and deep that is? That's not probably very easy to get to those areas to clear something out like that. So they probably would have that for life, maybe. So just wanted to point that out with hydroencephalopathy and stuff and what's kind of going on there.
That can happen, or can it happen in a particular brain also?
I would say it's probably possible for the interventricular foramen, but maybe not as likely because it's probably a little larger. So it might be less likely to clog up versus the cerebral aqueduct is pretty thin and small. So it's more likely that things could accumulate and clog that area potentially. And ideally it would never clog, but obviously it can happen. So I would just say it's probably less likely. I wouldn't say that it can't happen because, as you guys know, with healthcare, anything is possible. Even when you think you know everything and have seen it all, you'll get something strange that comes in and you're like, what the heck? How did this happen? And that's how some things get missed because people assume that can't happen. It wasn't in my textbook. All right. So there are two lateral apertures. There's one medial aperture off that fourth ventricle, and this is where your cerebral spinal fluid would leave the ventricular system and go to the subarachnoid space, which surrounds the brain and the spinal cord, so all the way down as well. And let's see, what else do I want to say? Yep, we already talked about encephalopathy. All right, so that was pretty cool. Cerebral hemispheres, or the cerebrum, you know, when they say, oh, you're very cerebral, or like cartoons will make jokes about that, they're talking about the thinking part of your brain, right? So cerebral hemispheres makes up the largest component of your brain at about 83%. So that's a big chunk of it. That's why it's so big and mushroomy looking. But also there are things that give it more surface area, just like on your cell membranes, right? When you had the microvilli that could give you increased surface area, we can do the same thing with our brain. So that's why it looks all funky. Have any of you ever at Halloween taken like a melon and like carved it out to look like a brain? I have. It's fun. You should do that sometime. And then they make these cool like zombie cupcakes where you can do the frosting like the brain. It's pretty cool. All right, so surface structures. So we have more surface area by things called gyri, which are the convolutions or the elevated ridges of the tissue. So like this is a gyrus, like this, right? The sticking up part. And then we also have sulci, or sulci, the shallow grooves between the gyri. So because we have all these sticking out parts and involutions that give us more surface area, thus more neurons, more neuron connections, more interneuron connections, that is what makes us higher level thinkers. Okay, so if you look at other animals that are more simple in their brain activity, they don't have these gyri sulci, or not to the degree that we do. So that's important for what makes humans humans. Okay, so where the neurons live would be the gray matter, right, where the neuron cell bodies are. And the white matter is where the axonal tracts are, because remember, axons had the myelin sheath around it. Myelin is the fatty tissue, thus what makes it look a little more whitish. So when you guys did like the frontal cut, the coronal cut, and you looked at it, you could see the gray and the white matter. Okay. What else do I want to tell you? There are a couple of areas that you will need to memorize. So we have a central sulcus. And that would be this guy right here, central sulcus. Okay, that's central sulcus. So that divides the brain into frontal and parietal lobes. And you guys can kind of notice that this sort of goes along with the the bones of the skull, right? Naming these areas. But if you overlap it, it's not like, look how large the frontal is compared to where your actual frontal bone is on the brain. Right, or covering the brain, if you will. So it's similar areas and named for those similar areas. And then parietal would be so frontal is this end of the brain, parietal would be that part, occipital is this guy back here, right? Then we have the parieto-occipital sulcus, so that's exactly where it's saying it's dividing the parietal area from the occipital area. Dun, 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 dun. And then you have the lateral sulcus, which separates the temporal lobe from the frontal and parietal lobe. So that would be this guy right here. Okay, that's your lateral sulcus. Cool? All right, then we have fissures. So these are deep grooves, and we only have 2 of them. And this might be where you guys had the really dense connective tissue that you called dura mater was found in this area. Okay. So you have a longitudinal fissure which divides the 2 hemispheres. That's sagittally, okay, sagittal. And then you also have the transverse fissure which separates the cerebrum from the cerebellum. So that would be down here, right? Okay, the other one was like along this line between the 2 hemispheres was the longitudinal fissure, then you have transverse fissure. All right, I'm going to erase all my drawings on there for a minute so we can come back to that. Bless you, bless you. All right, and cerebral cortex, so 2 to 4 millimeters thick. 40% of the total brain mass is called the gray matter. Remember, neuronal cell bodies. This is also where you see your pyramidal cells when you guys are doing your histology this week. I'm just saying. Anyway, the main control center of the body. So let me scroll up a little bit here. So cerebral cortex is the main control center of the body. Main control center of body. And this is our conscious mind. Our thinking part. We are aware of this and we're doing stuff because of it, and we make, there's an N in there, we make decisions about it, right? So that's what that's saying. So generalizations about the cerebral cortex. Each hemisphere controls the opposite side of the body. So right cerebral cortex controls the motor side of your left arm and foot and stuff, correct? Okay, and then vice versa. There are specific sensory, motor, and association areas or thinking, idea processing areas of your brain in different parts of the cerebral cortex, and the 2 hemispheres are not exactly equal. So for example, you have the idea of one side of your brain might understand the subtle meaning of a word in the text. Like you read the word, you understand it, but, and you have a concept about it, but then the other side of your brain might be the literal, actual part of it, right? So like right now, my 7-year-old daughter is very literal about everything you say to her. Okay, so she's using that side of her brain a lot more than the concept side on the other side, okay? And then I have my 9-year-old that's very conceptual. But Mom, that meaning could also be this. And that means that I can go do that now, right? No. Okay. So what I'm going to mention here, and no one area works entirely alone. You need connections and information across both sides, which we'll get more into next time we come together. But I wanted to just draw real quick your attention to these funky drawings here called the homunculus. homunculus. Okay, which basically means little man caricature. Okay, so if you guys do look at this online before you come in, because this isn't one of those pictures, but you'll see somebody that the blue side is talking about the motor part of it because it's located in the primary motor cortex or precentral gyrus, which we'll talk about next time. The peachy reddish side is talking about the somatosensory cortex, so the post-central gyrus. So these terms pre and post, same thing like with the neurons. Pre is on the before side of the sulcus. Post is on the after side of the sulcus, if you're thinking anterior to posterior, okay? And so basically with the motor, you have certain things like the hand that's really big, but like the arms are kind of skinny. And the face or head is kind of big, and the sensory stuff is kind of big for motor. I shouldn't say sensory. Scratch what I just said there. If you think about how many motor things your face can do, let's put it that way. Like you can smile, make all these kind of funky facial expressions and things like that, right? You can move your lips and your tongue in all kinds of crazy ways. Some people can't do it as well as others, like taco tongue and flip your tongue around and cloverleaf tongue. Anybody do that? I can't do cloverleaf. I can do taco tongue. Versus the peachy side, which is the sensory. Notice like the lips on this guy's face is huge, right? Because your lips are very sensitive. You have a lot of sensory neurons in this area. And the hand, you guys can do so much sensing with your hand, you don't even realize it, which we'll talk about next time. Okay, so lots of sensory with that. So I want you guys, if you get a chance, look up, Google homunculus, and you will see two funky, very looking different people. One's the sensory side, one's the motor side. And I want you guys to take a look at it so when we come back and talk about motor versus sensory, it'll make sense. And one guy has huge genitalia. Which one do you think that goes with? There you go. So take a peek at that. I'm thinking that we will, because you guys had asked not to do the extra lecture before, after that last exam, so you guys know that I will make a recording. I will try to get it done tomorrow-ish. So sometime over spring break or before I see you guys on Tuesday, watch that video. That way we don't get behind. I want you guys to come back and you can have questions about it. So we'll do a little review of everything or something. Because remember, our next exam is on a Tuesday, not on a Thursday like it has been. So I will make a recorded lecture. I will post it. I will try to remember to post the thing about that Get Body, virtual body for $1 thing. If you guys want to do that, that's pretty cool. And all right, we'll do some clicker questions.